This is part 2 of the Digestive System lecture. We are starting with chapter 24, section 10, the pancreas. So the pancreas functions as both an endocrine and an exocrine gland. So we will look at the endocrine portion of this gland when we get to the endocrine system chapter. So the endocrine portion is what's involved with the insulin and glucagon secretion and regulation of our blood sugar. And that involves a structure called the pancreatic islet. So again, we'll look at it then when we get to the endocrine system. The exocrine portion of the pancreas is what we are concerned with in the digestive system. This includes our structures called the pancreatic acini, and these structures produce our pancreatic juice. So this pancreatic juice is going to be secreted into the small intestine, right into the first part, the duodenum, via that hepatopancreatic ampulla. And that pancreatic juice is going to enter the small intestine and help digest our food and all of those molecules that have just entered from the stomach. So in pancreatic juice, again, just like all fluids of the body, we're going to find lots of water. We're going to find salts. We're going to find some sodium bicarbonate, which is a really important buffer since we have that strong stomach acid entering via that pyloric sphincter into our duodenum. So this hepatopancreatic ampulla is going to be situated just a few inches below that to help buffer that really acidic stomach acid so it doesn't damage your small intestine and doesn't inactivate any of these enzymes. And so these enzymes are going to be what are really important for digesting our food. So these enzymes, again, they're proteins. So these enzymes are very specific to the proteins to the molecules that they break down. So these are proteins, again, with those little active sites on them like we looked at in ANP1 that we talked about. This is where the action of an enzyme happens. So these enzymes, again, are going to be very specific to the molecules that they break down and are going to be very important. These enzymes are going to be specific to the molecules that they break down. So again, you're going to recognize some of these names like amylase that breaks down starch. We saw this in the saliva in the mouth. We also are seeing this now in the pancreas. So this is just, again, another amylase or enzyme that digests starch released from the pancreas. We also are going to have lipase. So we saw lingual lipase released from those glands under the tongue. This is pancreatic lipase. Again, kind of the same thing, going to digest those dietary lipids, usually in the form of triglycerides. So we're going to break those fats down into their individual pieces and then be able to absorb those. We also are going to see lots of enzymes that break down different proteins. There are a lot of different proteins in the foods that you eat. And so those molecules are very specific. So we're going to have a specific enzyme for those individual molecules. So that's why you'll see things like trypsin and chymotrypsin and carboxypeptidase. All of these things are going to just digest different proteins. Again, elastase. All different examples of different proteins that these molecules are going to break down. What's important is to see that these are all secreted as proenzymes. So again, just like we looked at pepsin and pepsinogen in the stomach, we are going to have trypsin and trypsinogen, chymotrypsin and chymotrypsinogen, carboxypeptidase and procarboxypeptidase. So again, these are going to be prohormones. These are going to be secreted as inactive, larger molecules that are going to be activated in the small intestine when they mix with those food molecules to digest them. That way we're not breaking down your own cells and your own pancreas. Pancreatitis is a really painful inflammation of the pancreas, where if these ducts here are major, so they're showing you this as the major duodenal papilla in this book, or in this picture. Your book calls it the hepatopancreatic ampulla. Just the little hole that lets the bile duct and the main pancreatic duct drain into the small intestine. If that would get blocked up by, say, a gallstone, these enzymes could back up into our pancreas and get inappropriately inactivated and start breaking down our cells and causing a lot of tissue damage, inflammation, and pain. So, common condition that you will see, again, gallstones are very common. They can get stuck anywhere along that tube. If they block off the pancreas from being able to secrete its digestive juice, we can see these digestive enzymes get activated inappropriately inside that pancreas. So the pancreas can digest pretty much every macromolecule. It secretes that amylase for starch. It secretes multiple enzymes for proteins. It secretes a lipase for fat. And it also is one of the only places that releases enzymes to digest our nucleic acids. So in food that we eat, there's still DNA and RNA if the things that we were eating were living. So we need to be able to break down those DNA and RNA molecules, right, into their sugars that we can use and into those nitrogenous bases that we can also use. So we are going to see these enzymes ribonuclease and deoxyribonuclease, again for RNA and DNA, to break them down into their respective pieces so we can use those as well. So that is pretty much all the pancreas does for digestive-wise. Again, that's all of those secretions in the pancreatic juice. We'll talk more about that when we get the pancreas when we get to the endocrine chapter. The liver is another accessory organ of digestion. So it is our largest internal organ, and it takes up most of the right upper quadrant, right underneath the ribs. So it's going to lie right underneath that diaphragm on the right side. And it's going to be made up of cells called hepatocytes. So anytime you see anything hepato with the prefix hepato, that is going to mean liver. So our liver cells are going to be our cells that help produce bile. So we looked at this when we looked at the circulatory system a little bit. We said we were going to have all of these portal veins. So this hepatic portal vein was going to bring in all of this blood from the digestive tract. So it was all of the blood that had drained and had received all those nutrients and molecules that your body had just absorbed before we sent it to the heart to send out to your entire body. So this hepatic portal vein is going to bring in that oxygen-poor, nutrient-rich blood and is going to drain that blood through these little hepatic sinusoids in between our hepatocytes. Those hepatocytes and macrophages that are in there are going to help filter and clean all of that blood out, absorb whatever it needs to, and then send the rest off to the hepatic vein to go back to the blood, to go back to your heart, to be oxygenated, and then sent out to the entire body. So these little hepatocytes are going to be really important to help form those correct little hepatic sinusoids to allow the blood to flow between them. While they're forming these little channels, they're also, again, producing bile. So they're going to be secreting bile flowing in the opposite direction as the blood from the hepatic portal vein into our bile duct. Again, these cells are just kind of dropping little individual drops out of each cell, are going to collect in what we call biocanaliculi, and they're going to be sent to bile ducts. Though, again, the bile ducts are going to drain into the right and left hepatic ducts and so on that we looked at in lab and we'll look at on the next slide. So those biocanaliculi, again, are just going to collect the little tiny drops of bile that each little hepatocyte is making and send them to bile ducts that are situated next to the hepatic portal vein. Again, the blood and the bile are flowing in opposite directions. So those hepatic sinusoids are going to be really important to allow the blood to get filtered and come in contact with our macrophages that are going to help get any kind of old blood cells, bacteria, anything foreign that the liver knows shouldn't be sent on out. Again, just really helpful to filter everything so we don't poison any parts of our body or we don't send any toxins to the brain or anything like that. We saw our portal triads in lab. So we have our bile duct, our hepatic artery, and our hepatic portal vein. A branch of each of those is going to be situated at the end of each, they're situated in a little group called a triad, kind of in each corner around a central vein. So you can kind of see this like hexagon-like shaped section of a liver lobule. And then these little hepatic portal triads here. We also look at the liver in different anatomical orientations, which are what we looked at in lab. Again, we had these hepatic acinus, which again connects central vein to central vein. And these are going to overlap kind of the areas that the portal triads are at. Zone 3 is going to be closest to the portal triad. So it is going to be getting all of the nutrients from the hepatic artery first. So zone 3 is going to be the first oxygenated and get all of the good stuff. As we move outward towards our central veins, which would be near our zone 1, we are going to have more damage in that direction. As for if we would have decreased oxygenation or anything like that, that's where we are going to have damage first, is around that central vein, and it would be damage last around our hepatic portal triad, where that oxygenated, nutrient-rich blood is coming in. So again, kind of just a different way that we look at the liver when we're looking at functional units. One is kind of an anatomical structure, and then that other way is kind of, it is anatomically oriented that way, but it's more of a functional unit. Again, just more of a vocabulary kind of thing. So what did we talk about with the liver? We looked at the flow of bile. We looked at the flow of blood. So we did both of these in lab. We practiced this. So we saw in those hepatic portal, or those hepatic triads, we saw a bile duct. All those little bile ducts, again, are going to collect from the right lobe of the liver into the right hepatic duct, and into the left lobe of the liver, from the left lobe of the liver to the left hepatic duct. Those two ducts are going to join each other and form the common hepatic duct. Again, hepatic means liver. So the common hepatic duct is coming down from the liver. The cystic duct is going to join that. So cystic means gallbladder, or kind of little sac, really. So the cystic duct is going to join in from the gallbladder to the common hepatic duct, and they're going to form now the common bile duct, because the common bile duct is going to have bile in it from the gallbladder and the liver. And you see bile can flow in both directions. If this sphincter right here, which we said are like little muscle doorways, they're going to close down really tight. If this is closed, meaning we don't have any need for bile in the small intestine right now, that bile is going to flow back upwards into the gallbladder, and it's going to stay in that little sac and get concentrated and kind of modified. So we're going to have a concentrated, more modified, stronger, if you want to think about it that way, form of bile from the gallbladder, mixing with this brand new kind of milder form of bile from the liver. And that's usually what the mix of those two is what gets released with the pancreatic juice into the small intestine via that hepatopancreatic ampulla or sphincter right here. So in this picture, you can see what ducts are in the liver, what ducts are in the gallbladder, and then how they're going to join from that with the duct from the pancreas, and that we're going to have that all of that enter the duodenum. So again, a bile stone or a gallbladder stone could block anything in this pathway and kind of block either secretions from the liver or the pancreas. Again, here is our blood flow through the liver. So again, we're going to have oxygenated blood from the hepatic artery and that nutrient-rich deoxygenated blood from the hepatic portal vein, all flowing into our hepatic sinusoids to get filtered and then flow into a central vein. That now filtered blood will flow into that central vein, get sent to the hepatic vein. The hepatic vein joins our inferior vena cava, and then our inferior vena cava dumps right into our right atrium of the heart, so our blood can get oxygenated and circulated again. The liver also has lots of other functions other than producing bile and filtering our blood. It is also responsible for a lot of metabolism of macromolecules. So carbohydrates, mainly glucose, is going to be able to be stored as glycogen inside the liver. And it's also going to be able to be broken down then once it's stored back into individual glucose molecules and released into our bloodstream. So if you think of each glucose as like a little Lego, and we stack them up into a big tower, that would be a big stack, that would be glycogen. And then we can take each one of those bricks apart, and those would be back to our individual glucose molecules. And again, we're going to talk more about that when we get to the endocrine system. So we also can do lipid metabolism. Your liver can also store fatty acids and cholesterol, really important. And we can also do some protein metabolism. A really important step of protein metabolism is removing our amino group, which is an NH2. That's a nitrogen and a hydrogen group. So we need to deaminate these proteins. That's just the term for removing one of these NH2 groups. So we're going to remove this. We're going to convert that molecule to urea, and then we're going to be able to excrete it. So this is really a key step for protein metabolism because this amino or this NH2 group is going to be really important for us to be able to remove, to be able to start breaking down our proteins. We also have talked about multiple times how the liver is really important to synthesize our proteins. So albumins, globulins, prothrombin, and fibrinogen are all proteins that we kind of briefly touched on when we looked at the blood chapter. So the albumins, again, are responsible for the osmolarity of our blood. So if you remember back to our net filtration pressure equation, we said that blood colloid osmotic pressure had to be at a normal limit to be able to reabsorb the fluid from the capillary bed, or your patient was going to get edema. So that's really critical to maintain accurate blood osmolarity, and then blood volume, and then blood pressure, right? We also looked at our globulins when we talked about our immune system, right? We said our antibodies are our immunoglobulins. They protect us from infection. Prothrombin and fibrinogen were involved in our blood clotting pathway. So this is going to be really important if you get cut, that if you have these proteins working correctly so that your clots will be able to form and you won't have increased bleeding. So really critical proteins that are in the blood formed by the liver. Again, your liver processes lots of drugs and hormones to make them at safe levels for you or completely inactivate them. We also excrete bilirubin, which we said can be toxic, right? If bilirubin builds up, it can go to the brain and be really toxic. The liver is going to make our bile salts, which again is going to be in our bile to help digest our fats. The liver is going to store glycogen, our fat-soluble vitamins, which are A, D, E, and K. It also stores our B12. And then our really important minerals like iron and copper, which we looked at how iron is stored in the liver in the blood chapter. We also saw in the last picture how those macrophages were able to do phagocytosis and filter out all of the things in the blood. And our liver is actually going to also help activate vitamin D. So we're going to absorb vitamin D from the sun and the sunlight into our skin, but it's going to have to be activated by the liver. So really important that your patient's liver is functioning correctly, or you can see the very long list of problems that they're going to start to have. So liver, very critical as an accessory organ of digestion, but also very important for many other functions as well. So definitely want to know what the liver does. I feel like you should know all of these functions really well because, again, liver disorders are very common. So when your patient comes in with those liver disorders, you want to start thinking immediately, what could possibly be wrong? What labs could be wrong? What symptoms am I going to start to see? What nutrient deficiencies are they going to have? Are they going to be at risk for increased infections and increased bleeding? What if we have to do surgery? Right? So start thinking about all these things. The liver is going to be really critical. So not only for digestion, but just really important that we discuss the liver here and that you read the section in the chapter and understand what your liver does overall as well. All right, so what about that little gallbladder? So the gallbladder, again, is that little they call it a pear-shaped organ. It's just a little pouch that sits below the liver. It is going to store and concentrate bile. Again, when that little sphincter is closed and there is no fluid flowing out of the pancreas and the common bile duct into the duodenum, that bile is going to be able to reflux back up into the gallbladder, and it's going to be stored up there. So it's going to be able to be stored and concentrated again, like I said, made a little bit stronger. So the gallbladder is going to have a muscle layer as well. It's going to have a smooth muscle layer, and it's going to contract, and it's going to push that bile into the cystic duct, then into the common bile duct. So the gallbladder is also going to be able to contract in response to different chemicals, right? Different nutrients in your digestive tract. So that's going to be really important for when your patients have pain. They're going to tell you with different foods, they have different pain. So that may be an indication of what's going on and what organ is involved, if that's an organ that's involved with digesting that nutrient. So what is in bile? What nutrients does it dissolve? Fat. Bile is what we use for fat digestion and absorption. So if your patient eats a large fat-based meal and they start to have right upper quadrant pain every time they eat that, every time they eat a fatty meal, but if they eat a meal high in carbohydrates or protein, it doesn't bother them so much, we might start leaning towards the gallbladder. So inside our gallbladder, again, we have our bile. Our bile is, again, a fluid of the body, so it's going to have lots of water. It's also going to have something called bile salts. Some of our bile salts here that your book lists are the chenodeoxycholic and cholic acid. These are going to work like dish soap. These are going to emulsify your lipid globules. So if you've ever washed dishes and you've had fat on any of the things that you've washed, you know you really have to put soap on your sponge and scrub it more, and you get lots of bubbles. It really helps break down that fat easier. That exact kind of thing is happening with these bile salts in your digestive tract. As everything's getting mixed around and those fat molecules are mixing with these bile salts, it's going to emulsify and break down those lipid globules just like the soap does when you wash dishes. We're also going to have cholesterol in our bile. We're going to have lecithin, which is just another protein. We're going to have some bile pigments, bilirubin, which again, you can see why this is green. Bile is usually a dark green or brown color because of that bilirubin in there. And we're going to have some ions, right? Again, some sodium, potassium, and chloride. We are going to have gallbladder our bile secretion be increased with any kind of meal, usually, but mainly with fatty meals, again, because it's what we need to digest fat. So again, really big clue of what could be going on with your patient if they come in and they have pain when they eat fatty meals, but are fine when they don't, when their meals are low fat. Okay, so now we are back on the track. We are back on the digestive track. So our small intestine, again, is going to be right below our stomach. It is going to start at that pyloric sphincter, which again is going to be one of those really tight muscle doorways that allows the stomach contents to empty into the small intestine. We said that the stomach contents was going to enter the small intestine slowly, because digestion and absorption takes a little bit, and we don't want just molecules just flying by and us losing them before we have time to fully break them down and absorb them. So our small intestine again starts at the pyloric sphincter, and it's going to end at the large intestine, and it's going to be about 16 feet long. It's going to have lots of little structures in there that help increase that surface area. So we are going to have 3 sections that we looked at in lab: the duodenum, the jejunum, and the ileum. And each one is going to have their own special features, again, that we kind of touched on in lab. So what is happening in our small intestine? Well, all of that chyme from the stomach is now mixing with our digestive juices. So all of the enzymes from the pancreas, the bile from the gallbladder, all of that. And it's going to get in contact with our mucosal layer of our small intestine. On that little mucosa, on that epithelial layer, we have something called a brush border. And there's some other enzymes on there to help us complete our digestion of our carbohydrates, our proteins, and our lipids. And if you remember, we talked about the carbohydrates and lipid digestion started in the mouth, but protein digestion didn't start until the stomach, while nucleic acid digestion doesn't start until the small intestine. It starts and ends in the small intestine because that's where the enzymes from the pancreas that we looked at, that ribonuclease and the deoxyribonuclease, are going to be able to come in contact with it. Again, the small intestine's main job is going to be to absorb things. This is where 90% of our nutrients and water is actually going to be absorbed. So it's going to be really important that we make sure we have enough time for all of those phases of digestion and steps to occur. The mixing of the enzymes is correct, and all of our secretions are present while food is in the digestive tract. So we can break all of those molecules down, and then we can have good absorption in our small intestine. So what does that look like? So again, Our small intestine is going to have that mucosal layer, that epithelial layer that touches the lumen that lines the lumen where the food actually is. It's going to be simple columnar epithelium. And again, we're going to see lots of specialized cells like we saw in the stomach. We are going to see lots of absorptive cells at the top of this villi. So we said we had those little finger-like projections called villi in the small intestine. And at the top of those villi, kind of lining the whole edge of those upper part of the villi, are going to be these absorptive cells. These absorptive cells are going to actually be able to, the cells that trap and absorb our nutrients. And you can see they're all very close to the capillary bed and the lacteal that are inside of that villus. So those capillaries and that lacteal are going to absorb our nutrients that we've just broken down and digested. The absorptive cell, you can see, has microvilli on top of it. They're like little mini finger projections. They're like little hairs that are hanging out, swooshing around, trying to catch all these little nutrients so we can absorb them. It's kind of just like if you've seen some coral in the ocean and like anemones, how they kind of just move around in the water and they have their little feelers out there and they just trap anything floating by and eat it. That's pretty much exactly what's happening with your microvilli. They're just kind of have their little feelers out into the fluid of the digestive tract and they're just sweeping around, kind of trap molecules in between those little hairs and then absorb them through that cell. We are also going to see a ton of goblet cells. So our goblet cells are going to be sprinkled all in here, and they're going to secrete our mucus, right? We said everything has to move through the digestive tract. It's very important that we have good movement through our digestive tract. So that we're going to need a lot of lubrication to do that, because as we move through the tube, things are going to start to get drier because we're going to absorb more water and more molecules. So these goblet cells are going to be all over the small and large intestine, and they're going to secrete mucus right on top of that absorptive cell and kind of spread it around and let everything move through that tube smoothly. We are also going to see some cells called enteroendocrine cells in the small intestine. Enteroendocrine cells, so entero means intestines, and endocrine is our endocrine system, so our hormones that we're going to talk about in a few chapters. So we have three different main types of enteroendocrine cells. We have our S cells, which secrete secretin. We have CCK cells, which secrete a hormone called cholecystokinin. And we have K cells that secrete this very long name hormone called GIP. It is called glucose-dependent insulinotropic peptide. This hormone has had about 7 different names in the time that I've known it. So if you see GIP, this is what they mean, even if those GI and P stand for different words because they keep changing them. So our K cells are going to secrete this GIP. So we'll talk about what these do here in a few minutes. So below our villi is still in that mucosa layer. We're going to have this lamina propria. It's kind of this extra tissue. So you can see it. It's right down here underneath this epithelial layer. It's kind of all the tissue that's up inside the villi underneath the epithelial layer where the blood vessels and the lacteal are. Inside of there, we're also going to find something called MALT. So we talked about this a little bit in the immune system. This is our mucosal associated lymphoid tissue. As you can imagine, we've said anything you bring in from the outside to the inside of the body can have a lot of germs on it. We have a lot of ways to combat those germs. We saw some enzymes in the mouth. We looked at the acid in the stomach. And now we are going to have a further step to kind of combat anything that would have made it this far, are these little extra lymphoid tissue nodules in the villi itself. So if anything would actually get absorbed, it would come right into contact with some lymphoid tissue and your body would be able to mount an immune response. This is also responsible for a lot of food allergies, right? That mucosa, that MALT is right there under the mucosa. If you eat something that you're allergic to, a lot of times this is where that lymphoid or that lymphatic reaction is going to occur. So we have a lot of inflammation and kind of things like that. Also a lot of autoimmune diseases will affect this lymphoid tissue, and you'll have a lot of issues with extra antibody production and that kind of a thing. So more to come on that when you get to those pathology courses. So underneath your mucosa, again, is going to be your submucosa, and that's where your blood and lymph vessels are, as well as your connective tissue. Again, anchoring this vessel down to our muscle layer, where our muscularis is. Again, we talked about our muscularis in the esophagus and the stomach. It's going to have those muscles in there that are responsible for peristalsis and actually move food through that small intestine. So we looked at some of our special features in lab a little bit. We said we had two really important ones. We had our Peyer's patches, which again were more lymphoid nodules. These lymphoid nodules are going to be located in our ileum. So our ileum is where our small intestine joins with our large intestine. And your large intestine contains lots of bacteria. Some good, some not so good if it gets any farther than in your large intestine. So, in that large intestine, again, or in that small intestine, right at the opening to the large intestine, we're going to find these Peyer's patches, which are these big purple lymphoid nodules here. Again, you can see they're right below those villi, making sure if anything gets in here, that reflux would reflux back from the large intestine, that if it came in contact right away with some lymphatic tissue. So again, those Peyer's patches are going to be really important in protecting your body from infection from any bacteria coming back in from the large intestine. We are also going to see Brunner's glands. Brunner's glands are duodenal glands that are going to be in the submucosa of the duodenum. Again, these are going to secrete a special mucus that has an alkaline or basic pH to help neutralize that stomach acid. So we said we were going to have that bicarbonate-rich buffer digestive juice from the pancreas to help with this as well. But again, that stomach pH is really acidic. So we really need to buffer it, and our Brunner's glands are going to help with that. So here's a nice summary slide of all of the special features that we find in the small intestine. So most of these we have talked about, but most of them, as you can see, are really all about increasing surface area for absorption. We want to have as much surface area as possible for those molecules to come in contact with and absorb our nutrients. We are going to have these little circular folds called pollicae circularis. They're going to be folds in the mucosa and submucosal layers, and again, going to help help increase our surface area and help move the chyme in the digestive tract in a spiral motion. So it's kind of hitting all walls of the tube. We're going to have our villi, right, which we said were our large finger-like projections that helped increase our surface area. And then our microvilli that we're going to also increase our surface area and help catch those molecules. They're going to also contain our digestive enzymes on our brush border. So our brush border, again, are going to be those little hairs on those microvilli. And these enzymes are going to allow for the final breakdown of nutrients before we absorb them. So we're going to look at a picture of this in a few slides. I think actually on the next slide. So we're going to look at a picture of this. What's happening is we don't want to lose any of these little tiny molecules. So we aren't going to break them down into their final form until right before we have the ability to absorb them, so they don't get lost because they're so small. So these brush border enzymes are going to help break down the nutrients immediately before they're absorbed into that absorptive cell, because those microvilli have caught and trapped it in there. We also said we were going to have those lymphatic capillaries and that are called our lacteals that are going to help absorb our lipids. So we're going to look at a picture of this in just a second. And inside of that digestive tract, we had intestinal juice. Again, it's going to be slightly alkaline, so we could buffer that gastric juice and have all of our enzymes be at that correct pH for their perfect function, right? Enzymes are proteins, so if they're kept at the wrong pH, they get what we call denatured. If it's denatured, it loses its structure. If something loses its structure, it loses its function, right? If we don't have the structure that it needs to be, like if your tire isn't that perfect circle and it goes flat, It doesn't function to drive your car very well. We need that optimal functioning. We want that perfect pH so that enzyme structure is the right shape and it's going to be able to function correctly. So we need to have those alkaline buffers in there to help get rid of, kind of neutralize that gastric pH. Okay, so here we can see some of those structures of the small intestine. We see those plicae circulars, those little folds. And then inside of those folds, again, is going to have our mucosa layer that has these cells in it. So here are these absorptive cells. These yellow ones here are our absorptive cells of the small intestine. Again, they are going to have the microvilli, which are these little mini finger projections. And on those microvilli, we are going to have a brush border. The brush border is enzymes. It's an enzyme brush border. So these enzymes are going to be located right here to break these molecules down to their final form so we can absorb them. So this is going to be really important so we don't lose any of these. So they get stuck right in here and we can absorb them through the cell. And if it's a carbohydrate or a protein, right into that blood capillary, right? So here's our villus. Here's one villi. The nutrients are going to be floating around right above that little finger projection. They're going to be absorbed to these little yellow orangey hepatocytes, absorptive cells in our intestine. And so those absorptive cells are going to see they just kind of pull these nutrients right through the cell. And right underneath them is going to be a capillary bed and a lacteal. The proteins and the carbohydrates, so you can see here our amino acid, which is our monomer or smallest unit of protein, and our monosaccharides or monomers of our sugars, our individual sugars like glucose and fructose, are going to be able to be absorbed right into the bloodstream. They go to the liver, right? Hepatic portal vein. They're going to be filtered by the liver, and then they're going to go out to the systemic, all over your body, systemic circulation, for cells to use them. Fat can't do this, right? Fat and water repel each other. Again, if you've ever washed dishes, you've seen this, right? You can't just use water really to wash fat. It doesn't come off. You have to use soap to break it down. So we said your body had kind of a method to do that as well with our bile. It was going to help emulsify and break down those big triglycerides that you ate into their individual fatty acids. So we are going to absorb those little fatty acids into our intestinal cell right here. This absorptive cell is then going to reorganize those pieces that you broke down back into that triglyceride. So you eat triglycerides, you break them down, you absorb them into the intestine as these little tiny pieces of fatty acids and monoglycerides. And then you reorganize them inside the absorptive cell back to the triglyceride, and then you put it in a little pod or on a little raft if you want to think about it like that. Again, this is called a chylomicron. So this little chylomicron pod of fat is going to be able to be absorbed into the lacteal. Again, remember lymphatic plasma is kind of a lipid-based plasma versus blood plasma is a water-based plasma. So proteins and carbohydrates will go great into that capillary bed and be filtered and be taken care of. The fats will not. They don't want to go into the watery plasma of the blood. So they're going to be absorbed into the lacteal in the form of a chylomicron. And then again, you guys know we talked about this in the lymphatic system. We reabsorb this via that lacteal to our lymphatic trunk, right? It's going to go back into the thoracic duct near the heart, right? And we're going to put it back into the circulation and all of that jazz, right? To be circulated and used by all of your body cells. So this is what happens with all of the food that you eat. Everything that you eat, everything that you're going to give your patient per oral, right? That they're going to ingest via their mouth is going to go through this pathway. We are going to start that carbohydrate and protein breakdown, or carbohydrate and lipid breakdown in the mouth. We're going to start that protein breakdown in the stomach while we continue to break down those lipids and carbohydrates. Stomach's going to empty all of that chyme, all of that it's broken down of the proteins with the acid and the pepsin and our carbohydrates and our lipids into the small intestine. The small intestine is then going to buffer that pH with our pancreatic juice and add some more enzymes to keep breaking down the food. Liver is going to bring in the bile to help break down our fats. And then as we move those molecules through the small intestine, they're going to come in contact with these microvilli and brush border enzymes. The final step of digestion will occur where we break these molecules down to their simplest forms and we absorb them across those absorptive cells in the small intestine villi. We then absorb them into our bloodstream or our lymphatic system, and we use those molecules all over the body to nourish your cells to have them do whatever we need them to do. So, everything you eat goes through this process, and everything you eat that we've now used and absorbed is going to be absorbed in the small intestine. Everything else is going to go to the large intestine. And so that's where we'll go here in just a minute. Here's a summary of the mechanical digestion that occurs in our small intestine. We are going to have two main types of movements, one called segmentations, one called migrating motor complex. Segmentations are local mixing contractions that occur right where the food is. So where the chyme is at, we are going to need to kind of mix the digestive juice and the food together. We have to get them together. So we are just going to have little segments, a little section of the small intestine kind of contract and relax, right? Contract and relax to mix things together. So once we've absorbed most of our meal now, so we're going to do this in the small intestine, right where the food is, kind of mix everything up, start breaking it down. Once we've absorbed most of what we've broken down, these little mixing movements will stop and peristalsis will start. So we'll start being able to move the leftovers into the large intestine. This is in the form of a migrating motor complex. So these are large peristaltic contractions that are going to move your chyme forward through the digestive tract. So we need to move through the duodenum and then the jejunum and then the ileum and then into our large intestine. So these migrating motor complexes are going to move food slowly every 90 to 120 minutes forward. The food is going to be in your small intestine for about 3 to 5 hours after your meal. So that's how long it's going to take for us to finish finalizing digestion and complete our absorption. So for about 3 to 5 hours and a couple migrating motor complexes every 90 to 120 minutes, we're going to move that food through all the stages of the small intestine. So those are our mechanical actions. Here are our chemical digestion. So again, most of this is going to be via enzymes. Talked about lots of enzymes already. You can see our enzymes that are going to be in our small intestine, again, are going to be those pancreatic enzymes, like our amylase and our peptidases and our lipases. Our small intestine is also going to have, again, lots of these final step enzymes. So you see this sucrase is breaking down sucrose into glucose and fructose. So sucrose is literally just a tower of 2 Lego bricks. It's if you took 2 Legos together or you stuck 2 blocks on top of each other. That's it. That's sucrase. We still can't absorb that. We have to break it down into one individual little Lego or block, if you want to think about it like that. And so we have to have these final enzymes like sucrase and lactase and maltase that are, again, very specific, all for only 2 sugar molecules that we are cutting into their individual molecules. Important if your patient has a deficiency of one of these, right? Lactose intolerance is due to a deficiency of lactase, which cuts milk sugar, which is called lactose, into its individual pieces, glucose and galactose. So people who are lactose intolerant don't have lactose and they cannot eat the sugar lactase, or they're lactase intolerant and can't eat the sugar lactose, because they can't do this step. So important that you know that sucrase is going to break down our sucrose molecule into glucose and fructose. Most of the sugar that you eat every day is a compound sugar. It's sucrose. And so sucrose is a combination of glucose and fructose. Lactase, again, breaks down milk sugar, which is lactose, into glucose and galactose, which are individual monomers or monosaccharides. And then maltase breaks down maltose into glucose and maltotriose. Again, just monosaccharides. The end goal from all of these, form monosaccharides for absorption. We cannot absorb disaccharides or anything larger, okay? So any more than one sugar molecule at a time, we cannot absorb. So that's where we're going to have these little brush border enzymes so we can break these sugar molecules down into their very small individual pieces right before we absorb them. You can see we're going to have some more peptidases that are going to break down our proteins, and we're going to have some more lipases again to help finish breaking down our fats. Again, some more nucleic acid enzymes to finish breaking down those nucleic acids into their final sugars, phosphate molecules, and then those nitrogenous bases. So, again, final steps of my molecule breakdown is going to occur on that brush border via these enzymes. Again, and now we're going to be able to absorb them. So here's kind of a summary of what we talked about earlier. Monosaccharides are going to be absorbed, again, via individual molecules are going to be absorbed via active transport or facilitated diffusion. Again, just depending on what the sugar is. Proteins are going to be broken down all the way into amino acids, and they're going to be absorbed again into our bloodstream. Our lipids cannot do this. Like we said, they have to be transported in the small intestine. They get broken down. Those triglycerides get broken down into their individual pieces and transported as micelles, absorbed into that absorptive cell, and released or reformed into triglycerides as a chylomicron, and then absorbed into that lacteal. The small intestine is also really important for fluid balance. It's going to absorb most of your electrolytes, vitamins, and water. So again, you can see most of what we bring in is also going to equal what we ingest and secrete is also going to be what we absorb, and very little of that is going to be excreted. So overall, we shouldn't have too much net water loss just from the production of our body fluids as long as we are ingesting enough liquid. All right, so here is a summary of everything that happens for digestion in the pancreas, liver, gallbladder, and small intestine. So really helpful kind of summary table. All right, last step in the intestinal tract is our large intestine. So the last stage of the digestive tract really involves just the production of feces and kind of final reabsorption of water. So we are going to be able to we have to move things from that beginning of our large intestine all the way out to the rectum. So we're going to see some peristaltic movements. A really important feature of our large intestine is having the correct bacteria. So it's kind of like a symbiotic relationship where you have good gut bacteria that live in your large intestine and help you kind of absorb anything that you can't absorb or create anything you can't. And we said this was a lot with our vitamin K. Our gut bacteria was responsible for making a lot of our vitamin K. We are going to have our final absorption of water, some ions, and some vitamins. We're going to have our formation of feces and our defecation reflex, which we will look at. Again, going to be really important to eliminate all of our waste products and none of the toxins or anything that is bad for us that is in there builds up in our body. So really important that we make sure that our patients are having normal bowel movements. So what does our large intestine look like? We are going to have these really big crypts or glands that have lots and lots of goblet cells. So when you look at the large intestine, you see lots and lots of white usually because you see these big goblet cells just full of mucus because, again, we're going to have that final reabsorption of water. We've already absorbed the majority of water in the small intestine. We are reabsorbing even more now in the large intestine, so things are going to get very dry. And so we're going to need increased mucus to help move things through there. Again, we're going to see some absorptive cells to absorb some water. We have no circular folds, no villi, but we do have some villi, some microvilli, again, on our absorptive cells to help trap any water molecules. we are going to see two types of kind of structural features that are unique to the large intestine. One of them is called our tennae coli. These are large bands of longitudinal muscles that allow for mixing in segments. So you can see this big long muscle that goes up the ascending colon, across the transverse, and then down the descending, that's going to allow different sections of the large intestine to move together. We are also going to see these little haustra or pouches that are formed when these little tennae coli contract. This is again where the mixing is going to occur and are going to help push things forward. So, what is occurring in the large intestine? We are going to have two important reflexes that help move things forward. As we have food coming into one compartment of the digestive tract, we are going to want to move the food that is in front, or anything that is left over in front of that area, out so we can make room for it. So we're going to see our gastroileal reflex and our gastrocolic reflex. Our gastroileal reflex is going to occur when we have food in the stomach. So if you just eat a meal, your stomach's going to stretch. That is going to stimulate the ileum to empty into the cecum, which is the first part of the large intestine, to make room for the new chyme that is coming in. So there's going to be new food entering from the stomach. We have to get everything that's in the small intestine out, so we're ready to deal with that. Our gastrocolic reflex is again from food in that stomach. So gastro, colic is colon. So the food in the stomach, stretch in the stomach, is going to stimulate the colon to empty. So we can move everything forward because we know we're sending the food from the small intestine to the large intestine. Well, now we need to send the food from the large intestine out so we can get all of that material that's in the colon out and make room for more. Haustral churning, again, is just that fancy term for when those little pouches are going to mix things together. And then we're going to also see that mass peristalsis, again, which is a large muscular contraction that's going to move things through the large intestine. So we're going to move things from the transverse colon all the way down to the rectum about 3 to 4 times a day. And again, usually after a meal because of this gastrocolic reflex. What digestion and absorption occur in the large intestine? Chemical digestion, again, that's not going to be from you. This is going to be from your gut bacteria. Really important. A lot of times that kids, they can digest bacteria that you cannot and help you get absorption or nutrients that you don't have or you can't get. Absorption, again, final place of water reabsorption. Very few ions and minerals or vitamins, but mainly just that last fine tuning of water reabsorption. So our defecation reflex, again, when we have to get all of our waste products out so we don't have any toxins or anything kind of building up. So we are going to send all of that material to the rectum, which we just talked about with that mass peristalsis movement. This stretch in the rectum is then going to stimulate these nerves in the sacral spinal cord region. This is going to trigger a parasympathetic response. So this is a rest digest feature, right? Who cares if those toxins build up if there's a tiger coming, right? In an emergency alert system. Nobody cares. We're trying to save your life. Well, now you're all relaxed and your body says, okay, let's deal with these other problems that aren't urgent, but are still very important. So that's the parasympathetic nervous system's ordeal. It deals with all of those factors in dealing with digesting and absorbing food and then producing like in urine and feces to get those waste products out of your body. Defecation is partially voluntary, partially involuntary. So the internal anal sphincter, again, so there's kind of like a double muscular door guarding the exit to the anus so we can make sure that everything stays in when it's supposed to. Again, incontinence is a big problem with a lot of patients. Maybe they have a sacral spinal cord injury. Maybe they have a brain injury that damaged the parasympathetic neurons. Again, voluntary and involuntary. So we have a lot of nervous system kind of crossing here, right? Normally something's involuntary or voluntary. You can choose to do it or you can't. We're going to have two muscles very close to each other. One, the internal one that is under involuntary control, being controlled by this parasympathetic nervous system response. And then the external anal sphincter, again, which is going to be under your voluntary control, which is going to have more of a skeletal muscle feature. Again, only going to be voluntary up to a certain point. At some point, this will if the involuntary signal is too strong, that external anal sphincter will relax on its own. So that defecation reflex, again, going to be important with a lot of your patients with spinal cord and brain injuries. You want to know where that's at because there are two different muscles here that have two different nervous system components controlling them. All right. So summary of everything that happens in the large intestine is here, table 24.6. And then the summary of everything that happens throughout the digestive system is in table 24.7. So the last thing that we are going to talk about are our phases of digestion and how this is all controlled and organized so all of these various parts work together in a coordinated manner. So the first phase of digestion is called the cephalic phase. Cephalic means head, right? So this is going to start really even before you eat. This is if you smell really good food, you see food cooking, you think about your favorite meal, or you start to taste your food. This is all going to activate these centers in the brain, and this is going to activate your facial and your glossopharyngeal cranial nerves to stimulate salivation, right? You know you can start salivating when you just get excited about thinking about food. This is also going to stimulate the vagus nerve to stimulate gastric secretions and get that stomach ready for food. So this is all the cephalic phase. This is just without you really even eating food, just thinking about it, seeing it, one of your other senses being activated and sparking this response of increased salivation and gastric acid secretion. Again, you can see there are 3 cranial nerves involved in this. If we have any damage to the brain where there are cranial nerves, maybe we're going to have a problem with this. All right, so what comes next? Well, you're actually going to eat food, right? So you got excited about it. Now you're going to eat your dinner. And so your gastric phase is now going to occur. This is going to have neural, so controlled by the brain, and also hormonal regulation, so controlled by hormones. So the neural regulation, again, is going to be stimulated by stretch from those stretch receptors in the walls of the stomach that we talked about with those other reflexes, and pH. When pH starts to decrease, we know we're going to have increased secretions and contractions of the stomach. We know we got food in there. We have stuff happening. So this is going to be stimulated by the nervous system. And then our hormones, our gastrin, again, that we talked about when we looked at the stomach, is also going to help stimulate digestive secretions. So the pancreas, the gallbladder, all of those secretions, those mucus cells in the small intestine, the Brunner's glands, right? All of these things now are going to start to be stimulated by gastrin, and we're going to get everything ready to have food. We have food in the stomach now. We know it's coming to the small intestine. And that's that's the next phase of digestion. So our intestinal phase of digestion is going to be regulated by our nervous system, again, by that enterogastric reflex. So if we have stretch, or this is a different reflex than we talked about. This is our enterogastric reflex. So if we have stretch in the small intestine, it is going to inhibit the stomach from emptying. Enterogastric, the small intestine is telling the stomach what to do. The small intestine is going to inhibit gastric motility and emptying because there's food in it already and it needs to deal with it. So we're going to have that digestion and absorption occurring in the small intestine, slowing the emptying of the stomach so we don't miss any nutrients. This phase is going to be regulated by those hormones that we talked about earlier from those enteroendocrine cells. If you remember, we had CCK and secretin and GIP. So we're going to talk about the CCK and secretin. CCK is cholecystokinin. This is going to stimulate the pancreatic juice to be released and contraction and release of bile from the gallbladder. CCK is what is released after you've eaten a fat-rich meal. That is what causes your patient's gallbladder to hurt, but they get that right upper quadrant pain. It's usually due to this hormone CCK. When we do studies in the hospital when we're looking at the gallbladder function, we administer CCK while we're imaging, and we can watch the gallbladder contract and eject bile, and we can see if it gets stuck anywhere. We also said one of your enteroendocrine cells releases secretin. Secretin stimulates pancreatic juice secretion and inhibits gastric juice secretion. So secretin says, hey, we need to calm down the stomach acid increase the bicarbonate and buffer this so we can increase our pancreatic juice secretion and those enzymes to be activated. So secretin is going to back up CCK. It's going to enhance CCK's activity and increase pancreatic juice and bile secretion, as well as inhibiting that gastric juice production so we don't have that super low pH. And again, here's our negative feedback loop that is showing you how this works. All right, and here's a summary table of those hormones that control digestion. Again, gastrin, secretin, and CCK. You definitely want to know what these do, what cells release them, and where they affect. All right, that is everything for the digestive system.